Welcome to today's Present Approaches to Testing webinar on molecular detectives, solving pathology puzzles with proteomics and mass spectrometry. I'm Dr. Linda Hasadstri, the Director of the Molecular Technologies Laboratory, as well as a consultant in the Division of Laboratory Genetics and Genomics here at Mayo Clinic in Rochester, Minnesota. And our presenter today is Dr. Akhilesh Pandey. Dr. Pandey is a physician researcher in the Department of Laboratory Medicine and Pathology at Mayo Clinic in Rochester, Minnesota, with joint appointments in the departments of molecular pharmacology and experimental therapeutics, as well as biochemistry and molecular biology. Dr. Pandey is also a professor of laboratory medicine and pathology, a professor of pharmacology, and leads the systems biology and translational medicine laboratory, as well as the mass spectrometry assay development efforts over in the advanced diagnostics laboratory at Mayo Clinic. Now, just a couple of reminders for everyone before we begin today. There will be a question and answer session at the end of the program. You can submit your questions online via the Q&A icon in Zoom at the bottom of your screen. Please only use the chat function if you have any technical issues. Today's program is being recorded and will be posted to our website in a few weeks. And with that, I'll turn things over to Dr. Pandey.
Thank you, Linda, for the introduction. Hello, everyone. Today, I'm very excited to share how proteomics and mass spectrometry can be used to solve puzzles in pathology and broadly in medicine. I have no financial or other conflicts. I'll start off by a very brief introduction to tandem mass spectrometry. Many of you have probably heard about this. And then I will talk to you about how formalin-fixed paraffin-embedded sections, or FFPE samples, can be used to look at proteins and metabolites. And the examples that I have chosen are examples where we already have published or we have stories in evolution to solve diagnostic dilemmas. I will also talk about the special user laser capture microdissection as one form of spatial proteomics, and I will end with how imaging modalities such as MALDI-based mass spectrometry imaging and finally infrared spectroscopy-based imaging can also be used to resolve diagnostic dilemmas. So tandem mass spectrometry, although sometimes it sounds like a complicated topic, but it's really a very simple concept. A mass spectrometer is nothing but an instrument that measures mass. And the reason it's called tandem mass spectrometry is because mass measurements are carried out in tandem. That means multiple rounds of mass spectrometry are performed. And this multiple round involves fragmentation of a selected ion. And I will illustrate this in the context of peptide sequencing by tandem mass spectrometry. It is also abbreviated often as MS. So on the left side, you see these cartoons of multiple peptides, which are present in a mixture. This is an illustration. Normally, from a complex mixture, such as tissue or cell sample, you have hundreds of thousands or millions of peptides. But here I've shown those 4 peptides where each circle represents an amino acid. From this peptide mixture, in the first round of mass spectrometry, one of the peptides is selected, and this is done automatically by the mass spectrometer. So one peptide is chosen within the mass spectrometer, and its mass is measured. So that's your first round of mass spectrometry, MS. In the second round, this selected peptide is subject to what is called collision-induced dissociation. Essentially, it's a controlled collision with an inert gas, which breaks apart each peptide molecule. Remember, even though the cartoon is showing you one peptide, it's many molecules of the same peptide. So each peptide molecule is fragmented somewhat randomly at the peptide bond, but in a controlled fashion so that it splits into 2, an N-terminus containing fragment and a C-terminus containing fragment. And on the right side here, I'm showing you the fragment ions that contain the C-terminus. And as you can quickly see from this illustration, this fragment here has one more amino acid than this fragment. So the mass difference should tell you what this amino acid is, if it has a unique mass, and so on. So basically, you're building these fragments, so masses of the fragments, so you can deduce the actual sequence. Here, I'm showing you the peptide sequence. This is normally obtained after the experiment, but this is quickly to visualize how you have these C-terminal ions, and what the mass spectrometer is measuring is this mass, this mass, this mass, this mass. And what it strictly measures is mass to charge ratio, but that is not the point of discussion today. Essentially, we can deduce the mass. So these are the masses of the C-terminal fragments, and these are the masses of the N-terminal fragments. And just by subtraction of one from the other, you can deduce what that amino acid or a modified amino acid might have been. So this is essentially what a mass spectrum looks like. And many years ago, people would sit and calculate the mass differences between these peaks to be able to say, oh, this is W for tryptophan, this is threonine, this is cysteine, this is glycine, and so on. But today, all of this is done automatically. But I want to point out that we know we can have amino acids that are modified, so post-translation modifications. You've heard of phosphorylation for sure, acetylation, protein methylation, ubiquitination are some of the other common post-translation modifications. So in the sequence, we don't just have a tyrosine here, but we have a phosphorylated tyrosine. And the mass difference between this ion and this ion corresponds to a phosphorylated tyrosine. So not only can we identify naked peptides, that is, peptides with no post-translation modifications, but also those that carry post-translation modifications. Today, the actual pipeline for a proteome-wide analysis is somewhat different. So I showed you how one peptide you can deduce the amino acid sequence, but often we start with a protein mixture. This can be from tissues, cells, dried blood spots, or FFP samples. And in most forms of mass spectrometry that is practiced today, we are actually not looking at proteins, but looking at proteins that have been digested with a protease. So we are looking at peptide mixtures. And one of the commonest proteases that is used is trypsin, which cleaves them into 10 to 20 amino acid long peptides. And it is this peptide mixture that is then injected into the mass spectrometer. And lots of data points that correspond to the peptide masses and their fragment ions are collected. And the way we identify peptides today is by searching it against a database, and then we get a list of identifications. So we identify the peptides, and as you can imagine, we know the human proteome now. They can be mapped up back to the proteins, so we know which proteins we are dealing with. So mass spectrometry is a very useful tool in the clinical diagnostic labs, and clinical mass spectrometry is not a new entity. For example, it's a very useful tool for amyloid typing, which amyloid is nothing but the deposits of misfolded proteins. And this deposition of proteins can lead to functional defects in many different organs, for example, kidney, heart, and others. And the way it used to be diagnosed is that you would basically look at the morphology, the way it looks under hematoxylin and eosin staining, but then you can do Congo red staining and then look under polarized light. And based on this special birefringence, people could conclude that, yes, we have a protein that is deposited. But there are lots of proteins that can be deposited. How do you identify which protein that is? Well, mass spectrometry is your answer. You can basically get these proteins out of these FFP samples, and I'll come back to this, and then identified by mass spectrometry. And in fact, Mayo Clinic introduced the world's first clinical mass spectrometry assay for amyloid typing in 2008, and it is currently the gold standard for finding out which protein is getting deposited as this generic term that is used, amyloid. There are other proteinopathies. For example, membranous nephropathy is caused by immune complex deposits. And again, there are many different proteins that can get deposited in the kidney. The commonest antigen is PLA2R in about 60% of cases, and this is very, very new. Mayo Clinic has developed a mass spectrometry assay for 13 different antigens, 7 of which were discovered at Mayo Clinic itself. So again, this is clinical mass spectrometry here and now. Finally, mass fix assay is a way to monitor monoclonal antibodies, for example, in multiple myeloma. And this assay is mass spectrometry based. It's a very sensitive and accurate assay, was also developed here at Mayo Clinic. But now I'm going to give you other examples that are pushing this boundary further into other types of diagnostic dilemmas, because now we know, I like to point out that tandem mass spectrometry is what a DNA sequencer is to nucleic acids. If you don't know what you have in hand, well, you can use next generation sequencing or NGS, which has a DNA sequencer built in to figure out what nucleic acids you have. Similarly, if you don't know what the protein is, you got a tandem mass spectrometer as your friend. So we are going to go through some of the applications, for example, for biomarker discovery in Wilson disease. Now, Wilson disease is actually a disorder of metabolism where copper is deposited in liver. And you could imagine, well, this is very easy to diagnose, but still on liver biopsy specimens, which is what you have, the patient doesn't come saying, oh, I have Wilson disease. So you have clinical symptoms, liver biopsy is obtained, and then the liver biopsy can have many different types of appearances, and they're very specialized assays that are available for looking at copper, but they have lots of issues related to sensitivity, cost, turnaround times, and they're also not available. So here we were challenged by our colleagues at Mayo Clinic to discover mass spectrometry based to use mass spectrometry based approaches to discover potential biomarkers where there is a suspicion of Wilson disease on liver biopsy samples on FFP slides, but could we find a protein signature, and with the hope that these proteins could then be turned into IHC type assays. So even though mass spectrometry is used as a discovery tool, the actual test could be an immunohistochemistry based test. So I won't take you through the primary data where we looked at many of these cases of liver biopsies from Wilson disease and compared them to many other liver disorders, and we are able to find proteins that were upregulated or had higher expression in liver samples from Wilson disease. This is metallothionein. It's a family of proteins, 1M and 1G, in patients versus controls. So in some of the initial studies we did, remember I just mentioned that our eventual goal was to turn it into an IHC-based test, and here we saw the biggest difference in expression in metallothioneins. So metallothioneins, they bind metals, and in this case, they are getting upregulated. The pathophysiology has not been worked out, but here we are using this as a surrogate to identify those cases where differential diagnosis with Wilson disease is being considered. And just based on H&Es alone, the other types of diagnosis that it could be confused with may be autoimmune hepatitis or other biliary tract diseases such as steatohepatitis. So on the left, I'm showing you that there are many different patterns in IHC where we have very high expression of metallothionein, and on the right side are controls. So basically, again, by IHC as a tool, we can tell them apart. But this was our pilot study, which was published as a discovery study. But our collaborator, Rondell Graham, here in Department of Laboratory Medicine and Pathology, he's a GI pathologist, and he collected even more cases and in a blinded fashion established an IHC for metallothioneins as a diagnostic marker, as a differential diagnostic marker for Wilson disease. And this validation study has now been published, and this is available as a diagnostic test. So again, going from a mass spectrometry-based discovery, we are already at a point where we have a new diagnostic marker by a very simple technique, IHC, which is available worldwide. So this test now can be deployed anywhere in the world. I'll tell you about another example, which is evolving as we speak of fibrolamellar carcinoma. So fibrolamellar carcinoma is essentially, it's a rare subtype of primary hepatocellular carcinoma, common in young adults, but again, it needs to be differentiated from the typical hepatocellular carcinoma. It does have a different clinical course, but its molecular pathology, the part that is known, is also very different. The major molecular basis of fibrolamellar carcinoma that is known is that there is a fusion event that happens where there is a deletion, a large deletion between this upstream gene DNAJB1 and protein kinase A catalytic A subunit PRKACA, which basically juxtaposes this part with this part. And what this ends up doing is it creates a constitutively active fusion protein, protein kinase A. And this now leads to the prototypical fibrolamellar carcinoma on the right. On the left, I'm showing you normal hepatocyte, normal liver histology, and hepatocellular carcinoma. So here the major diagnostic dilemma is still between hepatocellular carcinoma and fibrolamellar carcinoma. And the diagnostic approach, again, is multifaceted. The first is, of course, H&E staining. You can order special stains by IC, serokeratin 7, and CD68. And then molecular testing, FISH, break apart FISH probe is used to look at breakage of the PRKCA. And based on all of these, the evidence is put together for establishing a diagnosis of fibrolamellar carcinoma today. But this is not what I want to talk to you about. The real diagnostic dilemma is that, yes, it is true that the classic fibrolamellar carcinoma that I'm talking about arises because of this activated protein kinase A, and that's because of that breakage and fusion. But then there is another set of cases that look like fibrolamellar carcinoma, and our goal was to figure out what is happening in those cases. So we started with the usual, which is fibrolamellar carcinoma, to compare it to hepatocellular carcinoma and normal liver to say, based on proteomic profiling, can we tell them apart? So here we have these are controlled normal liver histology. By now we are looking at a PCA plot of the entire proteomic profile. I'm not showing you mass spectrometry data. This is looking at the entire proteomic data to say, can we basically bin these cases? These are controlled, so normal liver. This is hepatocellular carcinoma, and these are the classic protein kinase A-driven cases. We also did phosphoproteomics. I told you, using mass spectrometry, you can look at proteins, meaning unmodified peptides, but also modified peptides. So we can look at serine, threonine, and tyrosine phosphorylation. And this is the pathway downstream of protein kinase A, where this is the activated protein kinase regulatory and catalytic subunits. And these are many of the substrates. And we found hyperphosphorylation of many of these substrates on the substrate motif of protein kinase A in these samples. But the issue was, what about this rare subtype that our colleagues here at Mayo Clinic wanted to try to get a handle on, where they had done the usual diagnostic workup, including FISH, and they could not see this protein kinase A rearrangement. So it looks like and smells like fibrolamellar carcinoma, that's by H&E, but it's negative by this sort of confirmatory test for protein kinase A gene rearrangement. So as I mentioned, this is still evolving as we speak. But here we are looking at, again, a heat map of the entire protein profiling. Each row is a protein. And what you can see is that these are the FISH positive cases on the left, and the ones on the right are many of these unknown cases, a subset of these non-PRKCA-driven fibrolamellar carcinoma. And we are doing additional studies to try to figure these out. But again, so that means, as you can imagine, there are probably many molecular reasons that result ultimately in fibrolamellar carcinoma. And we have basically so far discovered the tip of the iceberg, that's protein kinase A, but now we can try to subclassify additional cases, in this case based on proteomic profile, and maybe we'll have to go back to the genomic profiling of these cases to try to figure out what is happening. But this helps us establish that yes, these are truly different, something different is happening in these cases. I'll switch gears again and talk about another molecular entity called mucinous cystic neoplasms. These are rare cystic tumors, and they can arise in liver, pancreas, retroperitoneum, many different places, often in women. They are benign, but there is a subset that if they show high-grade dysplasia and signs of invasion, such as invasive adenocarcinoma, then they need to be viewed differently. And the defining histologic feature here is that they have ovarian type stroma. The supporting cells look like they're ovary-like. On the right side is normal ovarian tissue, and I'm showing you on the left the low-grade mucinous cystic neoplasm, or MCN, a high-grade, and then this is with adenocarcinoma. So the stroma is reminiscent of normal ovarian stroma, and people have been talking about potential production of estrogen or its metabolites, because these could contribute to tumorigenesis in ovarian epithelial neoplasm. So again, the biology comes from ovarian tumors, which of course produce estrogen, but when they are in the context of these mucinous cystic neoplasms, is this look that we have under the microscope that is like ovarian stroma, does it have anything to do with ovarian phenotype? For example, do they make estrogen and its metabolites like ovarian stroma does? So here our goal was, the challenge really was to see, given FFPE tissues, could we detect estrogen and its metabolites from these slides where we have mucinous cystic neoplasms? And again, remember, we're not talking about ovary here. We are talking about other organs, for example, pancreas, where they are which is a common site of these cystic neoplasms. And on the right, I've shown you mass spectrometry-based assessment of many of these metabolites, but we cannot detect them directly. We have to take FFPE tissues, extract the metabolites. We have to derivatize them chemically before they can be visualized easily in the mass spectrometer. So now we've just switched gears somewhat. I'm now starting to talk about mass spectrometer as a tool for looking at other metabolites, not just proteins and peptides. So here we call these general molecules, as in this case it's a hormone, but these are small molecules as opposed to peptides, which are considered large molecules for a mass spectrometer. So we can look at estrone, estradiol, and many other intermediates. And then if you zoom out and look at the actual pathway, you got many other intermediates. So what we did was we developed this assay, and ovary, I'm showing you the signal for each one of these. For example, this is estradiol, and we could detect it in ovarian tissue. And this ovarian tissue, we developed the assay on FFPE material. and we could see it in the mucinous cystic neoplasms of pancreas. And estrone here and many other hydroxylated metabolites in the pathway are actually easily detected in these cystic neoplasms. We have just published this, and I think the important thing to highlight here is that this is the first demonstration of detection of estrogen and its metabolites by mass spectrometry. So this is the most definitive way to say, yes, we know, and of course, we are looking at the actual lesions. We know that those cells are producing when they should be. Normal pancreas was a negative control for us. So pancreas does not make estrogen. But in these lesions, we can see active production of estrogen. And this lays the foundation for future studies so we can study the biology more. So we will now zoom in a little more and talk about laser capture microdissection as a tool for spatial proteomics. So laser capture microdissection, it was invented more than 25 years ago at the National Institutes of Health. So it is a technology that is here now. And I should point out that this is also used in the context of amyloid testing. So those samples are collected after laser capture microdissection, but here I want to take a deeper dive into how it is done. So essentially, you can take H&E or any stained section or unstained sections, and you can mark the areas using laser. So it's laser guided. And there are many different ways of doing it. In one of them, the FFP material is on a slide and it falls into a plate or a tube. And these are the two major laser capture microdissection scopes, one by Leica, one by Zeiss, that I use. And the trick to this is that the sample, which can be a biopsy, is put not directly onto a glass slide, but onto a polyethylene naphthalate membrane or pen membrane, which is put on a slide. So here you see the glass slide. This is the pen membrane that has been put on, and the actual tissue will go onto the pen membrane. And again, on the left, I'm showing you a vertical section of the kidney, unstained or stained. Both of them are fine. So they are onto the pen membrane, and this allows it to basically be dissociated. If it is directly on glass, it sticks too well to glass and it cannot be removed. So it is basically a trick that this pen membrane can come off glass, but tissue directly on glass cannot come off. And I'll show you the use of this technology for some explorations that we are doing in different cancers. In one of them, the first example is on gallbladder cancer. So many cancer studies that are done, whether we are talking about discovery studies or other types of molecular analyses, people often take tumor tissue. We all know that tumors are surrounded by their own microenvironment, and if we are talking about carcinomas, they're epithelial in origin. So if you want to collect actual tumor cells, you have to preserve the histology. So if they are already on a slide, this is an H&E stain section, we know that these are the, so in this case, I should also point out that we are trying to get a profile for some tumors by looking at what molecular alterations are happening in tumor cells. And the only way we can do that is by not just taking tumor and comparing it to, again, a gross material collected from, in this case, say, a normal gallbladder. We need to get the epithelium of the gallbladder. So here, this is a normal gallbladder where we can cut out, for example, on the right, here, this part was cut out, the glands are gone, so the epithelial compartment has been microdissected out. So we are looking at where the action is happening, where the cancerous changes are about to happen. This is normal epithelium. And we have collected them for downstream studies. Of course, you can do DNA and RNA, but what I'm talking about is proteomics. And this is now tumor. So now you got a lot more to work with, but you can again avoid the stroma and get again the tumor epithelial cells. So all those have been cut out. And I'm not showing you the details of mass spectrometry, but I want to show this big picture to show you the power of mass spectrometry today. So we are collecting very little material from these FFP slides. These are 10 micron thick sections put on pen membranes, subjected to laser capture microdissection. So we are talking about tens of thousands of cells, maybe 100,000 cells. And we have also done single cell proteomics. I'm not talking to you about that here. So here, from one slide, we could get enough material that we can identify 8,000 to 9,000 or so proteins from the normal epithelium. I'm showing you this pilot study we have done. This is normal epithelium, and this is cancer epithelium, nearly 8 to 9,000 proteins from each sample. And remember, this is human proteome. We have about 20,000 genes. We probably understand less than half of them. And 2 or 3,000 of them have not really been detected anywhere easily. So out of the 16 to 17,000 or so of human protein that we could be looking at, we already have more than a half that we can easily see from LCM dissected material. Gallbladder cancer again, so it has a poor prognosis, but we are talking about molecular pathology, personalized medicine. HER2 has been identified as a new target on gallbladder cancer. So one question that we wanted to ask was, could we identify increased expression of HER2? Because it's often by gene amplification in gallbladder cancer. And of course, HER2 was originally identified more than 2 decades ago as a target in breast cancer, where all the therapies and all the research has been really done. But it's now also a target if you knew which of these individuals had overexpression of HER2. It's an EGF receptor family member. It's a receptor tyrosine kinase, which has no ligand. So it's always activated, but then amplification of this is really what people are looking for. So we looked at the case records of these gallbladder cancer cases, and based on FISH data that we had, either it was negative or it was not assessed. And in one case, this case 39, we'll come back to this, it was equivocal. So it was doubtful. People are not sure. And again, immunohistochemistry, it's a great technique, but there's still a pathologist bias there. And there is discordance that has been reported across pathologists for almost everything. When we looked at our data, we were looking at outliers, which proteins are really highly expressed in these samples. And the cases that we had, 2 of them really stood out for expression of HER2. This case here and this other case. And these are the 2 cases that are highlighted in yellow. And we thought, this is great. We already have sort of a case report, because now the molecular testing was not done, and we can look at proteomic data. This is still discovery data. We're trying to look simultaneously at nearly 8 to 9,000 proteins in the proteome of these laser capture microdissected cells. And we can tell, and there was no high expression on the control, the normal epithelium. So again, this is a signal that is unique to those tumor cells. But then we did a deeper dive into the records, and we talked to other colleagues here. What we were not paying attention to is that there is also a special panel, but this needs to be ordered. There's a targeted NGS panel, and that targeted NGS panel correctly identified these 2 individual cases as those that had amplification of HER2. So it's a special molecular testing that was able to determine, and the case that was doubtful by IHC, by FISH, was now confirmed to be not amplified by this. So again, different methods, they have their own pros and cons. And we went back to this amplicon that can be amplified. So again, we don't know because HER2 testing was done, but we are looking at other genes in that are often co-amplified. And in one of the cases, we can see that CDK12, which is in this, the protein level is again very high, and it's a little on the higher side here. So this is on the right side is HER2 in those two cases, and CDK12 in those two cases is here. So again, we can, even though we don't have genomic data on the whole chromosome, we have panel data now that HER2 is amplified. We are using the proteomic data, we can start to say that we can find these outliers where one gene or a set of genes at the protein level are overexpressed and maybe driving something. We also did, remember, I think I showed this, that HER2 IC was not done in those cases. And HER2, when we look at by IC, is blazing in those two cases, number 7 and number 34. So, but then we did IC on all the other cases. And this is the case that was equivocal, number 39. This was shown by molecular testing as not amplified. Proteomics said that this is not high level, but you can see how it's a gradient, that this could sometimes be called, well, maybe. That's why it was labeled as equivocal. So again, I think we could easily tell number 7 and number 34 as those with HER2 amplification. And we are trying out this type of methodology in other cases where molecular testing may not have been done, maybe it's not feasible, maybe there's no panel, but we can do discovery studies and find what are the proteins that are super amplified, like we captured HER2 in this case. I'll change gears again and tell you how we are pushing up our proteomics, mass spectrometry-based proteomics, to look at just a few cells and still be able to go deep into the proteome. And now we are focusing on cytology samples in the context of biliary brushings. So cholangiocarcinoma often arises in the context of a predisposing condition called primary sclerosing cholangitis, where individuals with PSC can be 100 to 1,000 times more prone to developing that than average population. That's a very, very high risk. So, but PSC is not a malignant condition, but cholangiocarcinoma is a deadly cancer, and it can arise in the context of that. So people with PSC are often called, and they are followed up very vigorously, and endoscopy is done to look at what is happening in the lining of the bile duct. So through ERCP, cells lining the bile duct are scraped. That's what is the biliary brushing. And those cells are then analyzed by cytology, that's on the left. So looking at cells in isolation, and by FISH. And this FISH assay was also originally developed here at Mayo Clinic. Now, we have a big issue based on this. So by imaging in these individuals, you don't find anything. It's all hardly there, but then you want to find as it develops in these individuals who are being followed. So all you have is access to these cells. And often when something abnormal is seen, it could be called suspicious, but suspicious is not good enough. And even with FISH, and this is again, a variety of alterations are seen. It's not like there is one particular alteration that's driving this, but there are multiple abnormalities. And even after including all of those, the sensitivity is still 60%. That means we are missing nearly 30 to 40% of individuals who have cholangiocarcinoma. That's a very high false negative rate. And we want to try to bridge that gap. And how do we know that we are missing those 40%? Well, as these cases are followed up, they are the ones who had cholangiocarcinoma, even if it was not diagnosed, they're actually going to die of cholangiocarcinoma that was missed. So that's the diagnostic dilemma. It's a big problem. Although it's a rare tumor. So what we have developed is cytospins are normally done for peripheral blood, but here we are now doing cytospins of these biliary brushings. So this is as an experimental technique. We are taking bile plus biliary brushings, they all come together, and then we are putting them on a slide. And then we have these clusters of cells, and we can appreciate nuclei by DAPI staining, epithelial origin of cells, pan-cytokeratin stain, and EpCAM stain. So we know that these are epithelial-derived cells, so bile duct epithelium. And what we are doing is now, as our experimental studies, we are trying to cut out these clusters of cells using laser capture microdissection. And there could be anywhere from 100 to about 500 cells in these clusters. So we can cut out these and subject them to mass spectrometry. And in our initial study, so this is again studies in evolution, we want to do better than current clinical testing. That's the goal. So we have all of the data. For these 4 cases, one of them is a confirmed case of cholangiocarcinoma. The 3 are suspected, of which one is highly suspected. And I've just shown you the varied alterations based on FISH and the overall assessment. But at the end of the day, one of them is being called as cholangiocarcinoma. And we have taken multiple such clusters from each one. And each cluster is now here. There are 4 colors for the 4 individuals. Purple is the one, this one here, lilac, is the one that is the confirmed case. The others are suspected. Now, each one is a cluster. And again, now we have fewer cells, but we can identify thousands of proteins from these. And our goal is to see if we can find a signature of cancer that is in the confirmed case that may not be there in others. Looking at our usual markers, cytokeratin 19 and EpCAM, well, these are epithelial in origin, and we can tell that based on mass spectrometry data, but this is just confirmation to say, yes, we're on the right track. Here, what I'm showing is a correlation plot of all the protein profile of each one of the cluster against all the other clusters. And what you see is that there is one cluster here from this first confirmed case, but just track this case. The other end, there's cluster 2. They are from the same individual. So they are 2. Remember, each cluster perhaps may be clonal, but it does not have to be malignant because these are biliary brushing. So you may hit the area where there's cancer, but there's also normal bile duct epithelium. So there's one cluster here, there's one cluster here, and the others are these 2 cases, they're like each other, and then this is yet another case. So we are trying to figure out, do we understand the cancer cluster? MCL1 is on 1Q21, which can be detected by FISH, and we can see that at the protein level. Just like I showed you HER2, now we can see amplification in these cases. We had also developed an assay to look at some of the mutations at the protein level of KRAS. Here, we found 2 of these clusters have a KRAS. KRAS mutations are common in pancreatic cancer, but they can also happen in other cancers, and including cholangiocarcinoma, but it's a rare one. But here, this is really the smoking gun. At this point, we were not sure whether cluster 1 or cluster 2 in the first individual who was a confirmed case was the cancer. MCL1 was the smoking gun. It was probably this, but we were lucky enough to be able to get this mutant peptide from this cluster. So here we have a cancer signature. And these studies are ongoing. So again, it's very exciting that we can take these cells, we can go from the looks, that is H&E, that's cytology, FISH, and we can add other molecular studies. And there are efforts underway here at Mayo Clinic to basically take these cells and do additional profiling based on DNA and RNA. And for the final vignette that I want to tell you about, where we have used mass spectrometry and other technologies, crystal deposition diseases. Many of you have probably heard of gout. It's a chronic inflammatory disease. It has bouts of acute episodes and is basically deposition of these inorganic crystals in joint spaces. They can also precipitate in the synovium, and is the immune response to this, because these crystals don't belong there, that causes inflammation. And there are a variety of crystals that can get deposited, and gout is caused by deposition of uric acid crystals. Pseudogout is by calcium pyrophosphate. And the diagnostic dilemma here was two things. One is, can we identify gout accurately from synovial biopsies, and also to distinguish it from pseudogout? And the issue was that the conventional way is to look at it to see, do we see any crystals in H&E sections? But also you can use polarized light for looking at birefringence by imaging, again, microscopy based. And there's one major caveat here, that uric acid crystals are urates in gout. If you do the usual formalin fixation, so FFP is formalin fix, then this washes away uric acid crystals. You have to use alcohol for fixation, but it's almost a catch-22. You have to guess that that's what is about to be the case, and then you can use alcohol fixation. And you may not always see the crystals. So we use, I won't go into the details, another form of mass spectrometry called multi-mass spectrometry to basically take these FFP samples and to look for signatures of these crystals. But before that, we had to develop the assay. So we took monosodium urate crystals alone, calcium pyrophosphate, and we could say that this particular mass, because these are chemicals, we know what they are. So a mass spectrometer should be our best friend. We can tell, oh, that's where monosodium urate is, that's the signature. This is calcium pyrophosphate. So if we just put this on this slide and look at it using the multi-mass spectrometer, we can tell them. But we want to see them in the context of tissues. So at the top is the tissue section. In alcohol fix, alcohol preserves uric acid crystals. And this, we have pseudo-colored the image, multi-mass spectrometry imaging. Brown here is positive. So the entire, even though here pink means there is no crystal, it's lit up with uric acid crystals. And this is what we are looking at here. We are asking the question, where do we see this peak on this slide? Well, everywhere. And there is no pyrophosphate. That's that peak here. It's blank. So we can see in alcohol fixed samples which preserves uric acid. We developed an assay for pyrophosphate. Now we are looking at pseudogout. And this is this greenish blue color. And that's pyrophosphate signature. And it's negative for uric acid. This is H&E, where you cannot even tell much. So you couldn't tell much on H&E, but we can tell very easily that there's pyrophosphate everywhere. But the best part is the formalin fixation, which gets rid, there are these holes where it should have been. But then when we are looking at those slides, we can easily see uric acid. Why? Because mass spectrometry is very sensitive. So we can see this. This is the most trivial problem that you can solve with mass spectrometry. So again, I think we have a new diagnostic approach to not only detecting uric acid crystals, but we can also distinguish it from pyrophosphate. But then my last piece is that we have usurped ourselves by using another approach, laser-based infrared imaging. This is an infrared imaging microscopy platform where these are very, very preliminary studies where we can basically see chemical signatures. It's a way to look at chemical signatures, nucleic acids, lipids, proteins, everything all at once. But once you derive your signature for what uric acid looks like, then you can basically go over the slide and say, show me if I see it. And this is, again, a pseudo-colored image that says, yeah, we can see gout. So again, and this is very quick, and this is a microscope versus a mass spectrometer. So we are very excited, but I think this is the future of some of these approaches where they're going to take us. For example, we should be able to do these are papers from other teams looking at colorectal adenomas. Can we develop some sort of a predictive signatures for who's going to have an actual adenocarcinoma? Then to do some sort of subclassification of soft tissue tumors, sarcomas, very notorious. Many of them are very hard to diagnose, to distinguish different types of gastric cancers. And my favorite is this recent paper that came out where they basically found a target using spatial proteomics that was amenable to a JAK inhibitor. And we are very fortunate to have outstanding collaborators here at Mayo Clinic. These are all of the different individuals that we have worked with in the last 7.5 or so years. And this is my lab, and I'll be very happy to take any questions. Thank you.
Was on mute. I apologize. Thanks so much, Dr. Pandey, for that amazing tour. There is so much on the horizon, I feel like, in this space, and the possibilities are seemingly endless. And so while I give folks a moment to type in any questions that they have, I just wanted to start it off with one of my own, which is there's obviously a lot cooking in the coming years, but what are you the most excited about amongst the upcoming projects that you have on the docket?
Yes, so yeah, we are excited about many things, and I think we are doing both discovery and validation studies. And for example, we are working on trying to find, just like the example I gave in fibrolamellar carcinoma, can we repeat that in sarcomas? So fusion negative sarcomas as one. So, and you do a lot of molecular genetics and profiling. So you know that Mayo Clinic is very, very advanced. We have even RNA-seq for that. So we can do DNA testing, we can do RNA testing, but all of this is good. But are there those cases that are falling through the cracks? That's one. The other is, even if we cannot find using our proteomic approaches some fusion that may have been missed, can we do more accurate diagnostics in the context of sarcomas? We have active projects where we have sarcomas with different outcomes. So what is really good is these are all diagnostic pathology samples. So they're obtained at baseline. So is there something intrinsic to the tumors? And of course, you could do DNA-based studies, you could do RNA-based profiling studies. I feel proteins are actually very stable. They are preserved well in FFPE samples. So we can do predictions on outcomes. We are starting a collaboration on Barrett's esophagus, where again, can we tell who's going to get esophageal adenocarcinoma? Is it intrinsic to the tumor where some signature, most of the individuals will not progress? And again, you need to have large collections with follow-up data to make these dreams come true. So we are very excited about these. And also we are doing a lot of studies in multiple myeloma with these types of classification and prediction approaches.
Thank you so much. Question from the audience. So I'm glad that you mentioned RNA-seq earlier. And that question is, how well do RNA and proteomic expression quantification correlate?
Yeah, that's a great question. I think people have been studying this for a long time. And there was a time when you couldn't do proteomics with limited material. And people had published that RNA and protein don't correlate well. And you could find studies where the correlation coefficient would go anywhere from 0.4 to 0.8. So it depends on the study. In cell-based systems, they may correlate a little more. But basically, so the way I like to say it is more in terms of plain English, which is easier to comprehend than a correlation coefficient. For some, RNA and protein correlate. For others, it doesn't. And we don't have the predictive ability to basically distinguish the two. So the point that I was highlighting is now we have the opportunity to be sampling nearly 9 to 10,000 proteins from very little material. So I used to say early on, more than 10 years ago, because RNA-seq was already there, you could do RNA expression, just go do it. Don't subscribe to this RNA correlates or doesn't correlate. You can do the RNA, so do it. Often it correlates, and sometimes it doesn't. But now I still say the same thing, but now I say, because proteomics has become a reality and is here now, you should also try to do proteomics, because now for the 9,000 proteins that you can sample or 12,000 in other settings, you don't need to worry about what the RNA says. You have the proteomic data, subject to many caveats. So we could have hours of discussion on the caveats, because RNA gives you a lot of information, isoform, fusion, things like that. So it's not RNA sequencing. We are trying to find a few peptides that represent the entire protein. So if we have a very uh, minute or very specific question about proteins, there is a good chance that is still not answered by what I say when I say, oh, we looked at 9,000 proteins. So some things are still missing and are not discussed openly. So there are caveats to that.
Yeah, thank you for that. You keep mentioning too, and I love this also, again, predictive capability also. So how do you foresee coupling the work that you're doing at the wet bench, for instance, with AI, since we know the AI wave is upon us?
Yes, exactly. So this is beautiful. So in two ways. One is, right now, as you know, a lot of the AI, if we just say anatomic pathology, let's say that people are taking H&E images and they are doing image analysis, and digital pathology, that's one major branch of AI. And they're trying to find hidden things that human brain cannot see, cannot guess, but the computer will find it. What I'm saying in that setting is we should use other types of imaging, and the infrared imaging that I talked about, it gives you chemical signatures. And we have to remember, hematoxylin and eosin, they are dyes that bind in ways we understand and don't understand. And we look at the color interpretation. Pathologists over more than 100 years have been trained on that pattern recognition. But they are still signatures of dyes binding to things in the cells. That's what the computer will catch. But infrared spectroscopic methods, they look at chemical signatures, and you can combine them with other H&E type approaches. I think they'll be much more information rich. So that's one. And the other is, if we know where the pathology is, like I was saying, if we want to look at epithelial compartments, or if we think, oh, this belongs to a problem in the glomerulus, we could be doing cutting out laser capture microdissection, and we could do proteomics. And we are actually doing that in the context of atypical ductal hyperplasia and DCIS in the context of breast cancer, because many of them don't upgrade to invasive breast cancer, for example. So many of them are just that. They will stay as precursors, but others are more cancerous, or they turn out to be in the same individual. So we are basically taking the imaging approach and the LCM-based proteomic approach in those. So we are active in that, but I think everybody should consider these approaches, because the more signatures you add on, I think the better your prediction will be. We don't have to lay all of our eggs in one basket of it's only H&E or it's only DNA or only RNA. Many times people are just using one. So these multimodal approaches will continue to become popular.
Thank you so much. Yeah, I'm excited for what's to come next. So seeing no additional questions from the audience, then I just want to take a moment to thank you all for participating in today's Present Approaches to Testing webinar on molecular detectives, solving pathology puzzles with proteomics and mass spectrometry. If your question wasn't answered during today's session, or if you have any additional questions, please include them in the evaluation. You can reference the link in the chat to access the evaluation now. Laboratory credit is extended for this program, so upon finalizing your evaluation, you'll have the option to claim credit and print your certificate. If you registered for this webinar and joined by phone only, please email us at mcleducation@mayo.edu if you do not receive the evaluation. And as a reminder, this program was recorded and will be archived to our website in a few weeks. Thanks again for joining us today, and have a great day.
Thank you for participating. Please click the button below to complete the evaluation and obtain credit.